Welcome back. This video will be covering the very end of chapter 17. We're looking at a kind of a hodgepodge of topics as we move from transcription and translation to the construction of polypeptides. And keep in mind, it's not always as simple as just taking the copy of one gene out to the ribosome and assembling that protein. There's a lot of different things that take place. And one of the really interesting things that takes place is the way a cell can manufacture multiple polypeptides very quickly. And it's by using what we call polyribosomes. This is something that they've just kind of discovered in recent years as the electron microscope has, that technology has allowed us to get a view of this actually taking place in the cell. Here is a picture. The messenger RNA molecule is running between these multiple ribosomes that are all lined up to kind of take advantage of this translation process. So you would have multiple transfer RNAs, just hundreds of them, maybe even thousands of them, bringing amino acids to these ribosomes that are all lined up. And of course, the messenger RNA molecule is going to move from 5' end to 3' end, so it's going in this direction. And again, the whole point of this is just to crank out a whole bunch of polypeptides as the messenger RNA molecule worms its way through multiple ribosomes. And so that's kind of an interesting advancement that the cell has to take care of this stuff very quickly. Oftentimes translation, again, is not going to be sufficient to give us a functional protein. There's many different specializations that sometimes have to take place in order to get this protein to work properly in the cell. Sometimes the chain is modified after translation. Sometimes they have to cut out a chunk. In fact, that's how insulin is made. You start off with a huge protein, and once the protein is made, enzymes will come in and cut out a middle portion of that protein, and then the two ends will be bound together, and that becomes the insulin molecule. Kind of strange way of doing things, but that would be a good example of how the protein is not quite ready even after it's made by a particular ribosome. Sometimes you have to add something to it, sometimes you have to cut a piece off, sometimes you have to bring a couple proteins together, tie them together. So there is some variety in what's going on here. This protein folding and modifications is something that biologists have been working really hard to try and figure out in the last couple decades. During and after protein synthesis, this polypeptide chain is going to start to spontaneously coil and fold and bend into a three-dimensional shape. That may not be very easy for the protein to do on its own. It may take some help by different enzymes. Like I said, there might be an enzyme that comes in and cuts a part out so that it's able to fold more correctly into the shape that's needed. Maybe we have to bring a couple polypeptides together to form the subunits of the protein. And when this is done, it's important to keep in mind it can be done in two different locations. Remember, there are ribosomes that are floating out in the cytoplasm by themselves, and there are ribosomes that are attached to the endoplasmic reticulum. What's going on there? Well, there are a couple different things. For starters, it's safe to assume that if you are a ribosome that's just floating around in the cytoplasm, you're going to be making proteins that are going to be needed in the cytoplasm. I mean, that's pretty logical. Well, what if it's not needed in the cytoplasm? What if it's needed somewhere else in the cell? Then that ribosome is going to be attached to the endoplasmic reticulum. So that protein then can be inserted into this endomembrane system and secreted out in some new location in the cell, maybe sent to the endoplasmic reticulum so it actually can be packaged and shipped outside the cell. These ribosomes are identical. Okay, it's not like a cytoplasmic ribosome looks different, works different than an endoplasmic reticulum ribosome. They are the same. And they can switch back and forth from free to bound to the ER throughout the lifespan of that ribosome. It's important to remember, though, that every time we make a polypeptide, the synthesis always begins in the cytoplasm. So it's going to start there no matter what. If it is a protein that is not needed in the cytoplasm, and it's one that is going to be headed towards the endoplasmic reticulum, there is a signal that the polypeptide uses to tell the ribosome, hey, we need to attach to the endoplasmic reticulum. These polypeptides are using a signal recognition particle. It is a very small protein that is going to bind to the signal peptide, and then this SRP will bring the signal peptide to the ribosome, and the ribosome to the endoplasmic reticulum, like you see here. So let's say we get this messenger RNA molecule, it gets started moving through the ribosome, and immediately part of the code is telling this ribosome that this protein that we're making is going to be required somewhere else in the cell. So these first few amino acids that are coming off the chain are what we call the signal peptide. They're going to be attracted to this SRP molecule, and the SRP molecule will bind to a receptor protein in the ER. Once that happens, obviously we've talked about receptor proteins before, that is going to open up the endoplasmic reticulum, change the shape of that protein, so that now this polypeptide can be inserted inside, and once it gets to the end of that chain, it'll be cleaved from the ribosome and be free to start the process of getting it coiled up into the shape that is required for this protein to be functional. So there may be some additional stuff that needs to be done with enzymes. We may have to chop this up into pieces. We may have to add something to it. There's a number of things, like I said before, that can happen to this protein before it becomes fully functional. All right, now the last little portion of this lecture, or the chapter, is talking about mutations, and they get into some specific types of mutations that can take place when we're manufacturing polypeptides, and they're really interesting to talk about. A mutation, of course, is a change in the genetic material. Point mutations will be the first type of mutation we're going to talk about. These are chemical changes that occur with just one base pair of a gene. So instead of having like GTCA, it could be GTCG. And that one nucleotide being off can cause an abnormal protein to be produced. So this is a very, very important thing to keep in mind. Some of these messenger RNA molecules, or genes, that are being copied by the messenger RNA molecule are thousands of nucleotides in length. And one single nucleotide being out of order or missing, or for example, here is sickle cell anemia. This is a disease that causes red blood cells to collapse so that they become very inefficient in terms of grabbing oxygen from the lungs. And you can see what's going on here as you look at the DNA sequence of the gene, you look at the DNA sequence of the mutant gene, and then what would happen if we tried to copy that with messenger RNA molecule. We would be off by one nucleotide, which this codon codes for glutamine, and this codon codes for valine. That one amino acid that is out of sequence causes this protein to bend in a direction that it's not supposed to bend, and therefore the red blood cell collapses, and now you have a very serious disease to deal with. There are two different types of these point mutations. We have nucleotide pair substitutions, where instead of having that's not drawn very well, instead of having a CG, we substitute in an AT, and that would be potentially a real problem. The other type is where you have an insertion or a deletion. So in other words, you're supposed to have like C, G, G. What would happen if we inserted an A into this spot? That's potentially a much bigger problem, because now all the codons that follow, in this sequence, we're just looking at one codon here, but this messenger RNA molecule could be made up of hundreds of codons, and every codon now that follows is going to be off by one. This is what we call a frame shift mutation. That would be an insertion. Instead of an insertion, what would happen if we deleted? We took this G out, and now we have a series of nucleotides that follow that are all going to be shifted one to the left because we're missing this nucleotide. These are potentially what we call silent mutations. They have no effect on the amino acid that is produced because of the redundancy in the genetic code. These are the types of mutations that if you get one, this is what you want. Missense mutations, we'll show you some examples of that. They code for an amino acid, but it's not the correct amino acid. And then nonsense mutations, these are potentially the worst of the mutations. This would be sickle cell anemia here. Potentially this might not be a big deal. The amino acid that replaces the one that's supposed to be there could be similar enough that the protein still is able to bend and fold correctly and be useful, but potentially it's going to really damage that protein. Nonsense mutations, instead of having an amino acid that is coded for by a particular codon, you get a stop codon. And wherever that stop codon hits, you're done. So this protein is supposed to be 100 amino acids in length, but if that stop codon happens after amino acid number 12, you're done. So now we have a protein that isn't even close to what it's supposed to be, and that potentially is the worst type of mutation. And so in your book, I'm not going to go through all of these. I've kind of explained some of them already. Here's an example of a silent mutation. Again, you see that didn't change the sequence of amino acids. Here is a missense one where we have the wrong one in place, and here's a nonsense one where we are finished making our polypeptide way too soon. So you can look at some examples of those in the book. This is taking them one at a time. Again, the insertions and deletions that I mentioned, why are they so potentially more damaging? Because everything that comes after the insertion or after the deletion is what we call a frame shift mutation. Everything's off. Every codon now is going to be off by one nucleotide. If we push a nucleotide into the sequence that doesn't belong, or if we remove a nucleotide from the sequence, everything that follows is what we call a frame shift mutation. And here again would be an example of that you're looking at. By adding an extra A in here, you now created a codon that is a stop codon, and it's not supposed to be a stop codon. It's supposed to be TAA, which codes for lysine. Okay, so that would definitely be a bad thing. If we remove a particular nucleotide from a sequence, again, we are missing a nucleotide as part of the messenger RNA molecule, which now means that we're coding for a different amino acid. Instead of having phenylalanine and glycine, we now have leucine, alanine. Every amino acid down the road from that mutation is going to be off. It's a frame shift problem. All right, so again, this would just be another example. And what would cause stuff like this? Well, there's lots of things that could cause it. Radiation, different chemicals, different things that happen during meiosis, mitotic division, recombination, stuff like that. Things can get messed up, and those physical or chemical agents are what we call mutagens.